All right, so we're on to our second section of our genetics chapter. This is chapter 12 in the Raven book. The first thing we're going to talk about in this chapter is the principle of segregation. And so this one is one that in the beginning seems a little bit tricky when you first hear about it, but in the end, it's really not anything we haven't seen before. So the basic idea is this: if you imagine one individual who is producing gametes through meiosis, right? That individual has two versions of every single gene in its body. One of the versions it got from its father, one of the versions it's got from its mother. All right, so when that individual is producing their own gametes, some of the gametes are going to contain the version of the gene they got from their father. Some of the gametes are going to contain the version of the gene they got from their mother. So, in other words, the two different versions of those genes, the one they got from the father and the mother respectively, are going to be segregated into different gametes. So they're going to separate themselves into different gametes. Some of their gametes might contain a father's chromosome, the other gametes would contain the mother's chromosome. So essentially what's going on here is this diploid cell has created two haploid cells, or actually four haploid cells, but we're separating apart those chromosomes that we got from the two parents during the production of gametes. So essentially what Mendel discovered here was meiosis, is basically all this is saying. He's saying that there are some diploid cells that are making some haploid cells. We start off with cells that have two copies of every chromosome, and we end up making cells that contain one copy of every chromosome. Now, the reason why that's kind of interesting is that meiosis hadn't been discovered yet. Chromosomes were still too small to be seen with the naked eye. The light microscope hadn't really been, it had been invented, but wasn't really to the point yet where it could be used to see anything as small as a chromosome. So people hadn't discovered or described meiosis. You guys, having already covered chapter 11 in this course, have like infinitely more information about meiosis than Mendel did, because it wasn't a thing that was known to anybody back when he talked about these chromosomes moving around, which is, I think, kind of interesting. So the idea of a Punnett square is that this is a way for geneticists to try to predict the outcome of a cross between two different parents that have a particular genotype, right? So it is a biological tool that's used to try to predict the outcome of a cross. It's not a guarantee, but it is the likelihood of something happening. So we're going to go ahead and work through the cross that we talked about back in section 1 of the lecture online, and we're going to look at a cross between, first off, a true breeding purple flower plant and a true breeding white flower plant. So in this particular instance, we're going to use big P as our dominant allele to represent purple, and little p is going to be our recessive allele to represent the white flowers. All right, now, as far as what letter you choose to do a Punnett square, that is completely up to you, right? But my suggestion is when you're working out a genetics problem, working out a Punnett square, try to choose letters that look different if they're uppercase and lowercase. And letter P is not a good example of that, because unless you're really particular about how you write them out, it's very easy to make an uppercase P look like a lowercase p. So that is something to kind of, a word of caution there. I like to use just letter A or letter B because there's no way to mix those up, uppercase and lowercase. It's a little bit safer to go that way with it than other types of letters. So the true breeding white flower plant is little p, it's homozygous recessive. True breeding purple is big P, it's homozygous dominant. We cross those together and we're going to get 100% of the offspring are going to be heterozygous. Right? So if we think about this diploid condition up here, this diploid cell, if we want to think of it that way, some of the gametes produced from this cell would carry this little p. Some of the gametes carried from this cell would carry the other little p. But in any case, no matter how you slice it, every single gamete from this individual white flower plant is going to carry a little p. All of them have to have a little p. Same thing down here. This cell can only give up big P's to gametes, so all of its gametes have to carry a big P. Now, if we do that cross between a parent that produces all little p's and a parent that produces all big P's, the only possible outcome is big P, little p, heterozygous. So 100% of the offspring have to be big P, little p during that cross. Now, this is kind of what I was showing you a second ago, representing this visually. If the diploid condition is big A, then half the gametes carry the first big A, the other half of the gametes carry the other big A. If the heterozygous condition is the case, so this diploid cell here, half of the gametes are going to carry a big A, the other half are going to carry a little a, because again, these are haploid. They can only have one chromosome and therefore one copy of each gene on those chromosomes. So if we work out a Punnett square here, now if we go ahead and do our cross between the heterozygous F1 individuals, what we find out is this. So we've got a Punnett square, and so when you're doing a Punnett square, the first thing you want to remember is how many boxes the Punnett square needs to have. If you're doing a monohybrid cross, and if you remember a monohybrid cross is where we're looking at one trait at a time, the Punnett square should have 4 boxes in a monohybrid cross. These gray boxes are what I'm talking about here. So this is our monohybrid cross. It's got 4 boxes. The female's gametes are here. Half of them carry a big P. The other half carry a little p. And again, we're talking about the F1 generation. This is the hybrids now, big P, little p. For the male parent, half the gametes have to carry a big P, and the other half also have to carry a little p. So in other words, we have a 0.5% or a 0.5 probability or 50% chance of getting this allele into a gamete, and a 50% chance or 0.5 probability of getting this gamete into a zygote as well. So 0.5 times 0.5 is 0.25, or 1/4 possibility of getting little p, and therefore getting the recessive phenotype. Similarly, we have a 0.5 times 0.5, so a 25% chance of getting this outcome, big P, little p, heterozygous there. We have a 25% chance of getting this outcome, and we have a 25% chance of getting this outcome. So if we take this one plus this one plus this one, that adds up to 75%. 25 plus 25 plus 25 is 75%. 75% are going to carry the purple flower phenotype or show the purple flower phenotype, and only 25% are going to show the white flower phenotype. So that's where we get our 3-to-1 ratio. And again, like I said back in our first lecture section, the 3-to-1 ratio only happens when you have 2 parents that are heterozygous for that trait. If the 2 parents have any other genotype at all, then the ratio or the expected ratio in the offspring is going to change. So this is the overall kind of big picture of what we did. We had our true breeding white and our true breeding purple. We bred those together, and we got 100% of the offspring that were heterozygous for that particular flower color trait. If we pick out one of these and let it self-fertilize, big P little p times big P little p, we have, again, 75% here, which are purple, and 25% here, which are white. So that is our 3-to-1 ratio in the F2 generation. Now, of course, the natural tendency here is to think about how this might apply to us as humans. And we might start thinking about how maybe siblings or parents or grandparents have certain traits and how those traits might be passed on to offspring. It turns out that there really aren't a ton of human traits that are quite as simple as what I've been showing you so far. There are some that are controlled by a single gene and that are following a simple dominant recessive type relationship. There's a few of those, just to kind of rattle off a couple of them. I've got a table here in a second that will show you some other ones, but some of the really simple ones are hairline. So if you have a straight hairline across the top of your forehead, that is one particular trait. If you have a widow's peak, where your hairline comes to a point in the front, that is the alternative allele for that trait. And widow's peak actually is the dominant form. A straight hairline is the recessive form. If we look at earlobes, so kind of a random trait, but your earlobes, if they attach directly to the side of your head and there's nothing kind of hanging down from that, that's what we call an attached earlobe. If you have an earlobe where it attaches to the side of your head and then you've got a little flap that hangs down below that, like most people do, right? Earlobes that hang down a little bit further, that is a dominant trait in that case. Right-handedness and left-handedness, believe it or not, is actually a simple dominant recessive relationship, which can also be affected by the environment. So if you were brought up, if your natural tendency was to be left-handed, but somebody sort of forced you to become right-handed, that is a factor of your environment. And so, something else to consider here is that we are more than just the sum of the genes that we carry. We also have environmental influences that make us who we are and what we are for a lot of these traits. The environment plays a big role in our phenotype and in a lot of cases. So that's something to consider as well. If we look at another trait, this one's called mid-digital hair. This one's really easy to see for yourself on your own body. If you look at your fingers, all right, so think about the tip of your finger where your fingernail is, and then go back one segment from there to the next, like between those two knuckles. If you have little hairs on your finger there, that is a dominant trait. You have what's called mid-digital hair. It's the middle of your finger, the middle of your digit hair. If you don't have any hairs, then you have no mid-digital hair. So having hairs there is a dominant trait. Not having them is a recessive trait. So all of those are fairly kind of like arbitrary things, like who cares if you have a widow's peak or not, or mid-digital hair or not, or whatever. They're not especially important in terms of natural selection, but there are others that are actually very important when it comes to natural selection. So if we're looking at humans, since we're not allowed as scientists or geneticists to take people and force them to reproduce and then count the offspring to try to figure out what the parents' genotypes are, right? That's not really a very ethical or legal thing that we're able to do. The only other alternative is to use what's called a pedigree analysis. And so pedigree analysis is it's a procedure that takes individuals that have mated presumably voluntarily and look at the offspring they produced, and from that try to figure out whether a given trait is dominant or recessive. So if you have a pedigree where we're looking at a dominant trait, you would expect to find that trait showing up in every generation of that family. Okay, so for a dominant trait, it doesn't skip generations, and a good example of that is juvenile glaucoma. This one actually is one that has a very, very negative effect on the phenotype in that it causes blindness by causing a breakdown of the optic nerve. This one shows up, if a family has it in their gene pool, it tends to show up in every generation because, again, you only need one copy of it to be expressed. You could have a big A or two big A's. Either way, you're going to have that juvenile glaucoma. The only difference is if you have two big A's, it sets in sooner and has its effects a lot earlier than if you just have one copy of it. So those kinds of traits are dominant. In terms of dominant things, we talked about mid-digital hair. Huntington disease, which is fatal, is one that's a dominant trait as well, but a really good example of the situation where dominant does not have anything to do with how common something is in the population. Huntington's, fortunately, is actually a very rare disease, but in the families that have it, it tends to show up in every generation because you only need one copy of it to be expressed. Essentially what it does is causes a breakdown of the nervous system, and from that, a series of really fairly horrible factors that eventually lead to death of people that have it. But dominant does not mean more common. PTC sensitivity, unfortunately for those of you who are not doing the class in person here, in lab, we actually do a lab, most of the time we have this lab in person, where we actually look for the sensitivity of a chemical called PTC, where we do DNA extraction and we amplify it, we make more of it, of your DNA, and then we actually eventually run a test to see if we can match up what your predicted phenotype is with what your actual phenotype is. Hypercholesterolemia is one where you individuals do not have the right types of cholesterol receptors on their cells. So when cholesterol is in their blood, they don't have a way to get it out of their blood, and so therefore it begins to build up in arteries and other parts of the body where they don't want it. Polydactyly, another good example. This is a dominant trait. Polydactyly is having extra fingers and toes. Now, most of us don't know people that have extra fingers and toes. So again, dominant does not mean more common. It has nothing to do with how common it is in the population. It just means that you only need one copy of it to be expressed. And if you have two copies, maybe instead of having 11 fingers or 11 toes, you have 12 of each or whatever. On the other hand, recessive traits, you have to have both copies of the recessive trait to see the recessive phenotype. So albinism works that way. Both red-green color blindness and hemophilia are what we call sex-linked recessive traits, which we'll talk about more a bit later on in another section, where the trait's actually on the X chromosome, and for that reason it tends to affect males much more commonly than females. Cystic fibrosis, muscular dystrophy, sickle cell anemia, all of those are conditions associated with recessive traits. If you are homozygous dominant, or if you're heterozygous, you are either going to have none of the ill effects of that particular condition, or if you do have the effects, they're going to be relatively mild and usually not life-threatening. If you're homozygous dominant, you're perfectly fine. If you're heterozygous, you may have like mild effect. If you're homozygous recessive, then you tend to have the full-blown condition, which in a lot of these cases typically is fatal. So this is a dominant pedigree. This is a good example of a dominant pedigree where we have a trait that shows up in every generation from grandparents up here, males and females, to the parents, again, males and females, and the grandkids. These happen to all be granddaughters of these individuals up here. Shaded means they have the trait. Open means they don't have the trait. So number one, we know it's not a sex-linked trait because both males and females have it. So that possibility is out if we're doing a pedigree analysis on this particular trait. We also know it's not recessive because there are no carriers for it. There's nobody that had the trait and passed it on, but they themselves didn't actually show the trait. That's actually a good way to find out that it's not a recessive trait, is if you don't see any carriers. In this case, either you have it or you don't, and because it shows up in every generation, we know it is a dominant trait, or dominant allele in that case. Now, albinism is a really good example, classic example of a recessive type allele. To be albino, you have to have a complete lack of the allele that you need to make melanin in your skin, in your eyes, in your hair, in your fur, your feathers, your scales, whatever other coverings you have as an animal. Other animals can be albino as well. So we have an albino rattlesnake in this picture, albino wallaby in the picture on the right with a normal joey here, normal baby here. So in this case, this female would have passed on a little a to the offspring, but the father passed on a big A to the offspring, and so therefore the offspring came out normally colored in that case, which is pretty typical actually to have, first off, albino offspring produced from parents who are not albino, and then going the other way, one more generation later, you have normal individuals produced from albino parents. So both of those two situations can be a bit of a shock. If you have two normally pigmented parents who produce an albino child, that tends to raise some questions when the baby is born. You could have two very dark-skinned parents, actually produce an albino child, which is typically pretty unexpected and may cause some questions to be asked in the delivery room. But this is again, it's a pigmentation thing. So it's in this case the amount of pigment and the ability to produce it at all as far as what the gene is doing. If you don't have the gene for melanin, you don't make any melanin. And so your skin is unpigmented, your hair, fur, your feathers, scales are unpigmented as well. Your eyes, everything is unpigmented. And so the eyes are red because in that case, what you're seeing actually is a layer of cells in the back of the eye, which contains blood vessels, and for that reason they appear to be red, as the result of seeing blood in the back of the eye essentially, but in blood vessel form. Normally people have pigmentation that is of other colors that sort of covers up that red. The red is there, but you can't see it because you have a brown pigment or a blue pigment or whatever in the front of your eye. Blue is a little bit different actually, it doesn't quite work that way. But brown or any other color aside from blue works by covering up the red pigment with a different pigment in the front of the eye. And there are albino birds and reptiles and fishes and all kinds of other things in addition to being the possibility of being albino if you're human. This one is a non-sex-linked trait. It occurs both in males and females in equal proportions. So we know it's not on the X chromosome. And for right now, take my word for that. But in the next section, we're going to get a bit more into what that actually means as far as sex-linked and X chromosome versus Y chromosomes. So again, in this situation, the most common occurrence is to see two parents who do not have the trait produce an offspring who does have the trait. So two non-albino parents produce an albino child, and then again, that albino child is perfectly normal otherwise, perfectly functional otherwise, and perfectly capable of producing their own offspring who is normal, non-albino, depending on the phenotype of their partner or their mate. So this is a recessive pedigree. And again, we know this one is not a sex-linked trait because it appears in both males and females. Both males and females are affected, and both males and females can be carriers for this given trait. It skips generations as well. So in this generation, neither of the two great-grandparents had the trait. In this generation, none of the grandparents had the trait, although 3 of them were carriers for the trait. In the parent generation, we actually have 2 individuals who have the trait, and then in the sort of current generation, we also have one with the trait as well. So this is a typically, fairly common type of recessive pedigree to find. So what we're going to look at next is a dihybrid cross. And what I want to do in the sort of tail end of this section of lecture is to just sort of define what a dihybrid cross is, and then we'll start going through the details of it when I start up the next lecture section, because we're getting close to that 20-minute mark that I normally like to keep my lectures to online. A dihybrid cross is where we're going to look at 2 traits in the parents that are followed through time. So instead of just looking at flower color, maybe we want to look at flower color and plant height. Or maybe we want to look at flower color and seed color, or seed color and seed texture, or we're looking at 2 traits at a time, right? Dihybrid means 2 traits being followed through time in the parents. All right. In this case, because we're looking at 2 traits, the Punnett square that we're going to look at is going to be a little more complicated, because we're not just looking at 4 possible outcomes, we're looking at 4 times 4 possible outcomes, which is 16 possible outcomes. So with a dihybrid cross, our Punnett square is always going to have 16 boxes, versus a monohybrid cross, where the Punnett square would only have 4 boxes. And if you're worried about this, we're not going to go beyond 2. Right? We're not going to go beyond a Punnett square with 16 boxes. We're not going to go look at trihybrid crosses or anything else, because quite honestly, if you take genetics, you wouldn't even do a Punnett square there, because you end up having a Punnett square with 64 boxes, and that gets to be kind of confusing after a little bit. If you're looking at just 3 traits, you have 64 boxes in the Punnett square, and that becomes pretty unmanageable. So we're going to keep it to dihybrid cross. We're going to look at an example here after I cut off this section again and start the next one, where we're looking at seed color and seed texture, I believe. And we're going to follow 3 generations of plants through a dihybrid cross to see how that trait might be inherited in the offspring. So with that, I'm going to cut off the section here, get it uploaded, and we will start up with our next section.